This is the last video in the viruses unit. In this video, we're going to focus on viruses that infect bacteria, otherwise called bacteriophages. You'll also hear these abbreviated as just phages, so I'll probably use those terms interchangeably throughout the video. Let's get started. No matter the cell type, the steps of viral infection are the same: attachment, entry, expression, assembly, and exit. However, complex-tailed bacteriophages handle the entry portion differently than animal viruses that we discussed previously. Here, you can see the tail fibers of the bacteriophage attaching to the surface of the bacteria itself. However, the viral capsid and protein structures do not enter the cell during entry. Instead, the virus itself injects its DNA genome into the cytoplasm of the cell. This works similarly to when you push the plunger on a syringe. The bacteriophage pushes down, putting its genome through the cell wall, through the cell membrane, and into the cytoplasm. Gene expression, assembly, and exit can then occur. Just like there are some latent animal viruses, there are also bacteriophages that can remain dormant. These are called temperate phages, and they are able to perform what we call the lysogenic cycle. The lytic cycle for a bacteriophage is the standard assembly followed by lysis of the cell for release of the virus. Interestingly, lytic viruses have been investigated as possible treatments for bacterial infections. Because they lyse the bacteria, they kill the cell. But their tail fibers are specific for attachment to bacterial structures, so they're harmless and cannot infect our human cells. With the emergence of antibiotic resistance making it harder to treat some bacterial infections, lytic bacteriophages may be the next line of treatment options, and in fact are used regularly in some Eastern European countries such as Russia and Georgia. If a bacterial cell is infected with a lysogenic phase, then the viral DNA actually becomes part of the bacterial cell chromosome itself. This means that every time the bacterial cell replicates, the embedded viral genome is also replicated. These embedded genomes are called prophages. Some of these prophages are important for bacterial infection because the additional new viral genes provide the bacteria different products. For example, you all have normal E. coli living happily in your GI tract that is not a pathogen. But there's also E. coli 0157H7, which is the E. coli associated with foodborne illness. The difference between the two is that 0157H7 contains a gene for the Shiga toxin, which is part of a prophage genome. When the bacteria picks up a new trait like this due to the prophage, we call it lysogenic conversion. This image will highlight the difference between lytic and lysogenic phages. In both, the viral DNA will enter the cytoplasm. But in the lytic cell, the viral replication proceeds as normal, leading to lysis of the cell. While in the lysogenic phage, we see the incorporation of the viral genome into the chromosome, and then the replication of that chromosome as the bacterial cell itself replicates. At some point in the future, eventually, the genome will pop back out of the chromosome itself and start to replicate and form new viruses. The assembly process will be followed by lysis of the cell. The benefit to the virus for incorporating it into the genome is that there now could be hundreds of infected cells in the population. This assembly would occur in all of them without the virus having had to infect each one of them individually. Instead, it just allowed the bacterial replication to make many more copies of itself before forming new virus particles. When working with virus in the lab, it's important to remember that viruses cannot grow on their own, and instead are obligate intracellular pathogens. That means in order to grow the viruses, you have to first grow their host cells. So if you're working with a bacteriophage, you need to grow the bacterial species that the phage infects. If you're working with an animal virus, you have to grow animal cells or use the animal itself. Often animal viruses are grown in either lab animals, cell lines, or chicken eggs. In fact, most of the influenza virus used to make the flu vaccine each year is grown in chicken eggs, which is why if you're allergic to eggs, you can't get that type of vaccine. There are now flu vaccines made from viruses grown in culture cells to avoid that problem for people. If you're growing viruses in a liquid cell culture, you'll see what we call a one-step growth curve. you'll notice that the initial number of virus particles actually drops down a little bit and then remains there for some time. This is called the eclipse period. It's during this time that the virus have attached to the cells, entered the cytoplasm, which is why you get the slight drop in number, and now they're going through the gene expression and assembly steps of their replication. After lysis of the original infected cells, the viral replication repeats itself as more and more cells become infected, and we see a rapid rise in the number of viral particles, called the rise period. Eventually, the number of particles is going to plateau and stop increasing because you've run out of cells to infect. All of the cells are now dead, and you no longer have any host cells for the virus used to replicate. If instead of liquid culture, you grow your cells on a solid surface like a Petri plate, you can actually observe the viral cell lysis. Here, the lysed cells can't grow on the plate, so we will actually get clearings where those cells have been killed by the virus. These clearings are called viral plaques, and they originally formed from one virus infecting one cell. So the number of plaques is equivalent to the number of viruses in your sample, and counting plaques is one way that we use to determine how many virus you might have had in a specific sample.